Hello and welcome back. In this section we'll be discussing the properties of biomolecules and how that reflects their fitness to the living condition. So macromolecules have a sense or a directionality. The way they're connected together matters, and we're going to look at that in a little bit. Macromolecules are usually informational, meaning they can carry information that's important for living systems. And biomolecules have characteristic three-dimensional architecture. These are not flat things. These are three-dimensional species, obviously, because we are three-dimensional creatures. And weak forces are what maintains the interactions that keeps biological structure and determines biological function. So biological structure is held together by weak intermolecular forces. So amino acids, as I'm sure you know, are the building blocks of proteins, and it matters how these amino acids are put together. They are put together from the amine end, or the nitrogen-containing end of the amino acid, to the carboxylate end in a polypeptide chain. So one carboxylate is connected to the next amine group in an amide bond, but one end, that means that one end of that polypeptide is going to have a free amine, and the other end is going to have a free carboxylate group. So generally the way we express this is by putting the amine end on the left on paper and the carboxylate end on the right. So it matters how these things are put together, and they go so they have a directionality or a sense that goes from the amine end to the carboxylate end. Polysaccharides are built up by joining sugars together, and it matters how these are put together too. So here's one example of a polysaccharide that is connected from the fourth carbon on the sugar backbone to the first carbon on the sugar backbone. So in other words, we have a 1,4 glycosidic bond, and we'll discuss this in more detail in a later chapter. The point of this is that polysaccharides have a direction or a sense. So in this case, the end that has the fourth carbon is connected to the end that has the first carbon, and this first carbon here is the anomeric carbon because it's joined to these two oxygens. So we'll talk about that again later on, but the fourth carbon then is joined to the first carbon, and so that is a 1,4 glycosidic bond. Actually, in this case, this is a 1,4 glycosidic bond. So the direction actually matters. And here we have some nucleic acids that are polymers of nucleotides, and so our sense here is that we have the 5' end. So this is the first carbon on this ribose sugar. So we have 1, 2, 3, 4, 5 carbons on that sugar. So this is called the 5' end. This is called the 1' carbon, and so this goes from 5' to 3'. So the 5' carbon end is connected to the 3' end of the next sugar. So that's our direction, 5' to 3'. All right, and so by having this directionality or this sense, these building blocks are able to be put together in specific arrangements, which gives them the ability to carry information. So they can actually carry information by having these little building blocks, these little pieces. In the case of DNA, DNA is made up of obviously adenine, thymine, guanine, and cytosine. And so these bases are arranged in a specific order in order to code for different proteins. And so here we see that this strand of DNA going from the 5' end to the 3' end over here is actually made up of the specific set of the nucleotides, and by having that specific set, it can code for specific, it can carry specific information to code for specific proteins. And here we have a polypeptide segment, which is a small protein, and it also carries information by having this specific sequence or arrangement of amino acids. And here we have a polysaccharide chain, which really doesn't carry any information because it's just a simple chain of sugar molecules put together. So it's just the same sugar molecule, glucose in this case, put together end to end. So this polysaccharide doesn't actually carry information. So these are all macromolecules, but the DNA and an RNA can carry information, and so can a protein, but the polysaccharide doesn't really carry any information. So the three-dimensional structures of these molecules are actually held together by weak intermolecular forces. So obviously covalent bonds are what holds these parts together. So we have covalent bonds holding DNA and RNA individual pieces together, and the protein individual amino acids together, and the polysaccharide sugars together are all being held by covalent bonds. But that would just be like a long chain, right? So what holds them into their respective three-dimensional shape? Like for DNA, we have a helical coil. For the proteins, we may have different three-dimensional moieties like alpha helix or beta sheet or something like that, which we'll talk about later on. And so what holds those things together? The weak forces, the weak intermolecular forces that are not the covalent forces. That's what holds these three-dimensional shapes. And these weak forces include van der Waals interactions, hydrogen bonds, ionic interactions, and hydrophobic interactions, which we're going to talk about now. So obviously proteins have this three-dimensional structure making up the overall protein. So we have that sequence, that list of amino acids that are covalently bonded together, but they're not in a long chain. They don't look like a road. They look like these globular species or something like that. They have some regularity to them that is three-dimensional, and that overall three-dimensional shape is held together by these weak intermolecular interactions. So here we have the antigen binding domain of immunoglobulin, which is obviously held together in this three-dimensional shape. So what are these weak interactions? Well, they're van der Waals interactions, which are hydrophobic type interactions. They are interactions that are caused from molecules bumping into each other and shifting the electrons, causing transient changes in partial charge on different ends of the molecule. So they result from nonpolar molecules bumping into each other and creating these temporary induced dipoles. Okay, well, all molecules have van der Waals interactions, even if they're polar, but we generally think of these existing in nonpolar molecules because they're really the only type of interaction that exists in nonpolar molecules. And so also we have hydrogen bonds, which are stronger than the van der Waals interactions. Hydrogen bonds result from a hydrogen being on a heteroatom atom like oxygen, sulfur, or nitrogen, and they are stronger than the van der Waals interactions, but their relative strength is proportional to the atom that is bearing the hydrogen. So oxygen-hydrogen bonds are stronger than nitrogen-hydrogen bonds. Okay, so we're going to look at an example of that soon. And next we have ionic interactions. These are fairly strong because they're fully charged. So we have like positive and negative charges holding these together. Now, they're still not covalent bonds, but they're fairly strong interactions, like from an ammonium group and a carboxylate group together. And then we have true hydrophobic interactions, which result from water displacing nonpolar molecules or nonpolar moieties in molecules and forcing them together. So water displacing these nonpolar species causes them to aggregate and results in hydrophobic interactions. So this is also a weak interaction. Okay, so van der Waals interactions are from, say, 0.4 to 4 kilojoules per mole. Hydrogen bonds, 12 to 30 kilojoules per mole. Ionic interactions are around 20 kilojoules per mole. And hydrophobic interactions are less than 40 kilojoules per mole. So we can see that the hydrogen bonds and ionic interactions are the stronger interactions of this list, with ionic interactions being the strongest. And these interactions all together influence the nature of biological structures. All of these interactions exist in biological structures. So biomolecular recognition is mediated by weak chemical forces. So molecules interacting with each other and recognizing each other, like a ligand in a binding pocket of a protein, that's mediated by weak chemical forces. And those weak forces restrict organisms to a narrow range of environmental conditions. In other words, people can't exist in 200 degrees Fahrenheit conditions because your proteins will denature. You can't exist like that. And so that's why organisms have to exist in this very narrow range of environmental conditions where their biomolecular processes can continue. So here we have an example of how these van der Waals forces are important to biological interaction. So here we have the lysozyme, which is a bacterial enzyme, okay, and we have kind of a binding pocket here that is recognizing, or is recognized by, an antigen binding site of an antibody against lysozyme. And so we see kind of a binding pocket where that antibody is attaching to the lysozyme, which is an antibacterial enzyme trying to deactivate it so that it cannot be active against that particular bacteria. So these intermolecular interactions are optimal at a certain distance from the atoms from each other. So as the atoms reach that optimal distance, we have a minimum energy. But as the atoms get further and further apart, that interaction becomes weaker and weaker. And as the atoms come closer together, as their van der Waals radii come closer together, the electrons begin to overlap with each other and repel each other, and so the energy increases. So we have this minimum energy that exists at an optimal van der Waals radius between these atoms. In other words, the atoms want to exist at a certain distance from each other, and that minimizes the energy. So that van der Waals radius for a hydrogen atom is like 0.1 nanometers, carbon is 0.17, nitrogen is 0.15, oxygen 0.14, phosphorus 0.19, sulfur 0.185, and then we have an aromatic ring, the half the thickness, because we're talking about the radius here. That means the radius is the distance, half the distance between two atoms, and so basically it's the distance of an atom. But half the thickness of an aromatic ring would be the van der Waals distance of an aromatic interaction, and we're also going to see some of those in biological systems, and so that's 0.17. So if you compare that to a covalent radius, it's much weaker because it's much further, and so covalent bonds are stronger, they're closer together. Van der Waals bonds are weaker and further apart. All right, and so here's some biologically important hydrogen bonds that we'll see. We have, so if a hydrogen is on an oxygen, then it's going to want to hydrogen bond to, say, another oxygen. That approximate bond length is 2.7 nanometers, or about, 0.27 nanometers, or about 2.7 angstroms. So oxygen, hydrogen, and then an O minus is about 0.26 nanometers. Oxygen, hydrogen, and then to a nitrogen. Okay, so an oxygen bearing a hydrogen that's hydrogen bonded to a nitrogen, 0.29 nanometers. A nitrogen bearing a hydrogen that's hydrogen bonded to an oxygen is about 0.3 nanometers. A positively charged nitrogen bearing a hydrogen that's hydrogen bonded to an oxygen is about 0.29. And a nitrogen bearing a hydrogen that's hydrogen bonded to another nitrogen is about 0.31. So all of these are about 0.3 nanometers. They're all about the same distance, really. All right, and so here's some good hydrogen bond donors and acceptors. The species that can be hydrogen bond donors and acceptors. The carboxylic acid can be a hydrogen bond donor. This hydrogen can be received by a partial negative on something else. Okay, and so here's an alcohol that can be a hydrogen bond donor. We have an amine, we have a primary amine and a secondary amine that can be hydrogen bond donors. And here we have some acceptors. Here we have carbonyl, we have ether type moiety, alcohol. Okay, and so this double bonded nitrogen is imine here, and we have this phosphorus oxygen double bond as well that can accept a hydrogen bond. So these guys over here on the right have a region of electron density that can accept a hydrogen bond, because remember that's going to be partial negative. And on the left we have the partial positive of the hydrogen that can donate a hydrogen bond. All right, so here we see an ion, or rather ionic interactions with magnesium interacting with ATP in this magnesium ATP complex, which we'll talk about later on in this course. And so this is an example of an ionic type intermolecular interaction. So it's not a covalent bond. It's not, the magnesium is not covalently bonded here. This is an ionic interaction. Here you can see ionic interactions contributing to the overall three-dimensional structure of proteins. We have this carboxylate end interacting with this amine end, and here we have an amine group as a side chain on a amino acid interacting with a carboxylate group. Okay. Oh, actually, that's what this is. This is an amine end interacting with a carboxylate group here. Sorry. And then we have these two groups. So notice one's positively charged, one's negatively charged, one's positive, one's negative. So they're interacting, the ionic species are interacting with each other. So these are ionic bonds in proteins, and they stabilize the three-dimensional structure of the proteins. So you can see here we have some three-dimensional structures of proteins here, and the idea of this slide is that these guys complement each other. So we have an antigen on the right, which is a small protein, a lysozyme from a hen egg, and we have an antibody molecule on the left that has a binding pocket that's structurally complementary to this antigen surface feature. Here we have the antigen, we have a surface feature in red that's going to bind somewhere on this antibody like a lock and key. So they're going to bind to each other like a lock and key because of the complementarity of the three-dimensional structures and the features that exist within the protein. So we have some positive and negative charges, or we have some, we may have hydrogen bonding interactions, ionic interactions, hydrophobic interactions, something here, and I'm not really that familiar with this complex up close, but if we looked at it in some modeling software, we'd be able to identify the types of interactions that exist here on this protein. Okay, and so this complementarity is what guides these many biological processes, like a lock and key. Biological systems are complementary to other biological systems so that they can fit together. And if a protein is denatured, it's lost its overall three-dimensional structure because something has disrupted those intermolecular forces in the molecule. So something's disrupted the hydrogen bonds or the ionic bonds or something like that. So what could do that? Maybe soap, adding soap, like if you wash your hands, that's why you can get rid of bacteria off your hands. Or if you heat the protein up, or if you change the pH, maybe you add a salt solution. Any of those things can denature a protein and cause it to lose its shape. And if it loses its shape, then it loses its function. It's no longer able to do what it was designed to do in that biological system. All right, thanks for watching, and I'll catch you next time.